Hello, everybody. This is Margot Buckles. I'm a volunteer with the Washington State Beekeepers Association, and tonight we're presenting Ask a Washington Beekeeper. And our guest is Molly Quade, who is a graduate student at Washington State University, and she is going to be speaking about entomopathogenic fungi for Varroa mite management. Basically, that's a fancy way of saying bug killing fungi. Molly began working in the bee program at Washington State University as an undergraduate, where she assisted on a collaborative project with Paul Stamets, investigating the role of Ganoderma resinaceum, reishi mushrooms extract in enhancing honeybee immune systems. This experience sparked her interest in insect-fungi relationships, and since then, she has contributed to a variety of projects focused on integrating fungal applications into commercial apiculture. Now a graduate student at WSU, Molly is researching the use of Metarhizium, I hope I pronounced that correctly, and other entomopathogenic fungi in integrated pest management with a focus on developing strains for Varroa mite management. She plans to continue her research in fungal strain selection with the long-term goal of advancing the application of fungi in commercial beekeeping and sustainable agriculture. And as an aside, Molly was the recipient of a scholarship award from the Washington State Beekeepers Association for this very important research. So Molly, go ahead and take it away.
Awesome. Thanks so much, Margot. So yeah, I'm really excited to share my research tonight with you all. Um, yeah, I'll go ahead and get started. But a little background. So I originally enjoyed learning about honeybees in the bee lab, but I really wanted to study fungi. And there are many reasons why people love and appreciate fungi. Maybe it's because they look beautiful, like these Amanita muscaria. Maybe it's because they're tasty, like these shiitake mushrooms here. Maybe it's because they bring people together, like how my friends and I here foraging for oyster mushrooms and morel mushrooms. Getting outside together is always fun. And a lot of people also appreciate fungi for the amazing ways that they've been implemented into medicine and just science in general, and the amazing advancements they've made. So, with that in mind, um, I was really interested in exploring how fungi could be used to help honeybees. So that's what led me here in the Hopkins lab. And here's a little outline of what I would like to cover today. So I'll give a quick overview of the stressors that bees are currently facing, just so it can be understood how these fungi play a role with helping the bees. And then I'm going to give an overview of Metarhizium with its use in integrated pest management, both past and current. And then investigating use of Metarhizium in apiculture. And then finally, I'm going to give an overview of my research that I'm doing as a graduate student. So first and foremost, if you're a beekeeper, you're already familiar with the fact that we rely on honeybees because they serve as a pollinator that we're able to manipulate as a livestock and basically make up for the lack of native pollinators that we would have that normally would be pollinating plants. But because we're growing crops in these large monocultural areas, and that's how our commercial agriculture is, we rely so heavily on honeybees because we're able to manipulate and transport them to different locations where we want to bring in pollination for our crops. So, whoops, plug in my charger real quick. So we know that in 2000, they estimated honeybee pollination, like the industry overall, to be worth $14.6 billion. But we know that that's obviously going to be a lot higher now. Just no one has crunched the numbers since 2000, but they should. So when we start seeing problems that honeybees are facing, it's concerning to everyone, not just beekeepers because we care about our bees, but also to anybody who needs to eat, which is going to be everybody, right? But this problem we're seeing of higher and higher mortality rates each year within commercial honeybee colonies, it's been getting drastically worse in recent years, and especially this last year was like the worst on record. Like I know a lot of commercial beekeepers were having like 60% mortality rates, and this is obviously very, very concerning. Well, what's causing this? Well, first and foremost, we know that bees go through a lot of different obstacles in order to survive and reproduce and continue living. So first of all, one of those obstacles would be collecting enough of a balanced diet of pollen and nectar. Oops, yeah, I wrote pollen and honey. What I meant was pollen and nectar here, to survive the winter. So you might see these beautiful frames of many different colors of pollen, and this would be an example of a very diverse source of food for these bees. So that's one thing. But then also bees need to have a large enough worker population to survive through the winter. So if you open a beehive up in the winter, which you should not do, you will see them all clumped together to keep warm. So that's another obstacle. But kind of the main one that is affecting all this high mortality is the fact that bees need to manage viruses, diseases, and different pests. And pests can, of course, include wax moth larvae, hive beetles, but mainly parasites, specifically the Varroa mite. And we know that the Varroa mite is very destructive to Apis mellifera, the honeybees we keep here in the US. And kind of the reason that is that it originally was a parasite to Apis cerana, the eastern honeybee. And just in the last 100 years, we've seen this host switch where Varroa mites started showing up on Apis mellifera, which is the one we have here in the US. And within a few years, it drastically spread all across Apis mellifera colonies. So because of that, it's led to decades of devastation, and beekeepers have to step in and perform some kind of management strategy to help bees not succumb to all the damage and vectoring of virus and diseases coming from these mites. So now not only are our bees facing the normal obstacles they might face naturally, such as gaining enough nutrition to survive the winter, having a big enough population to survive the winter, and then managing pests they already would. But now they're also dealing with all these other obstacles related to commercial agriculture, like losing workforce when they're getting transported during the day, and also neonicotinoids and other pesticides that they may be exposed to. And then on top of that, now they're having to deal with all these mites that are living on their bodies and damaging tissues as they feed, while also vectoring all these diseases and viruses. So with Varroa mites, we now have this kind of compounding problem where all these things stacked on each other have led to mortality, but mainly we know it's because of these mites. And of course, there are many different routes you can go to manage Varroa mites. So for one, you might rely on mechanical methods, such as using the powdered sugar method to manually remove Varroa mites from bees, or destroying drone brood, because we know Varroa mites are more likely to be reproducing in the drone brood, so we can scratch that out and kill a lot of them. However, those mechanical methods are either not always super reliable, because there's also going to be mites outside of the drone brood, or maybe that powdered sugar didn't get every mite. But more importantly, they're also going to be too time consuming to be implemented on a commercial level. So a commercial beekeeper is not going to be found out applying powdered sugar to their hives. It's just not what happens. And then we also have miticides from natural chemicals such as oxalic acid, formic acid, thymol. And of course, these can be applied in many different ways. And then there's also synthetic miticides such as amitraz, fluvalinate, coumaphos, and so an issue with these synthetic miticides would be they can build up residues in the comb, which are then going to be affecting the bees residually for long periods of time, with all the larvae and pupae that would then develop in the cells found in that comb. And then also all of these, the natural chemical and synthetic chemical miticides are going to require proper PPE and planning. And that doesn't necessarily mean not to use it, but just another thing to consider. However, the main issue we're seeing, that's the main important thing, is that mites are building resistance to these synthetic and natural chemical treatments. So with that, we're now seeing that these treatments are less effective, and that's a problem. And we know that this is because Similarly, with pesticide resistance developing with any type of insect or any type of pest, we often see this where, maybe a small subset of a population is able to survive some kind of topical treatment, but then they are able to reproduce and then carry that gene that has that resistance to whatever treatment they was applied on them. So then we end up with this second generation here that is now carrying this resistant gene. So over time, when we're using the same product over and over, or the same set of products, we see things like, for example, with these mites building resistance over time. So not only does this mean we need more management methods to alternate between to prevent that resistance from building. We don't want to keep using the same product over and over because that's how this resistance will happen. But on top of that, it would be ideal to find some sort of new strategy that would potentially be less likely to allow the mites to build resistance to it. If we could potentially have something maybe more sustainable. And with that, the idea of biological controls kind of comes into play. So if you don't already know, a biological control is, to put it simply, just using one biological organism to manage the population of a pest organism. So the common example with insects that I think most people would be familiar with is using ladybugs to manage aphids in your garden, where people will buy ladybugs at the hardware store and release them. And we know that because ladybugs have this life history of preying on insects such as aphids, that if we release them in the garden, they're going to prey on whatever aphids they find and manage them for us. So another example would maybe be using a cat to manage rodents on your farm or something like that. But because biological controls are maybe a viable option for finding sustainable management methods for mites, the main reason for that is that it's usually much, much harder for pests to build resistance to these biological controls. But other than that, biological controls are known for having low non-target toxicity levels. For example, if we release those ladybugs out, we know they're probably not going to prey on some other beneficial insect that they haven't shown history of feeding on. And they can also be easily integrated into sustainable and organic agriculture because we're using a biological, naturally occurring organism. So yeah, that mainly, to put it simply, a biological control could be a very good option for finding varroa mite management methods for this reason of the mites might be able to build resistance slower or not at all. And anything can be a biological control. So I gave the example with ladybugs, that would be an insect biological control, but they can really be anything: mammals, birds, bacteria, or even fungi. And so with that, I'd like to introduce Metarhizium fungi, which is kind of our star of the show tonight. And this fungus is entomopathogenic, just like we said at the beginning. So entomo means insect or arthropod, and then pathogenic means it is a pathogen, it infects. So entomopathogenic means it infects insects and arthropods. So this fungus naturally does that. And what's really fascinating about these fungi is that they're found in soils all over the world. And there's actually been a lot of interesting survey research done where they've gone out and taken soil samples of soils in all different regions and all different climates of the world. And basically, we know that Metarhizium and other types of entomopathogenic fungi are very much present in the soils everywhere. So hearing about these fungi that parasitize insects might seem like this really exotic idea, but in reality, these things are all around us all the time in the soil, unless you live in Antarctica. But other than that, you might be wondering, well, if they're in the soil, how are they infecting the insects? These fungi can also be saprotrophic, which means they feed on dead or decaying matter, organic matter in the soil. So they're not necessarily infecting insects. They can also just be in the soil decomposing things. And because of this, they're also considered beneficial because fungi, are our main decomposers. They're what allows us to not have a bunch of logs and everything hanging out all the time. Contributing to that soil cycle. So beneficial for that purpose. And it can live as an endophyte, which means it's known to be symbiotic with plants, but we're not going to get into that very much right now. So here's kind of what that non-infection cycle would look like. We start with conidia, which are spores, asexually produced spores. This is an asexually reproducing fungus. And these spores are dispersed into the soil or into the air. And they germinate and form mycelium, which creates a mycelial network, as shown here in this picture. And then that mycelial network produces more spores, more conidia, and then those spores are dispersed, and this cycle repeats. So this is the non-infective life cycle, and I don't really care about that because it's not relevant for us right now. But just so you know, it's able to do that. But what we really care about is this infective life cycle. So when that spore is released and begins its travel through the air, through the soil, it can continue in the soil like that and just continue producing mycelium in the soil. But it can also, if it has the opportunity, land on an insect's exoskeleton and basically start growing mycelium that breaks into the insect's body and grows from the inside out, feeding on the insect's organs and stuff. And I'll show this a little closer in a second. But basically, the main takeaway here is that these fungi are amazing because they're able to flip-flop between either of these life cycles. They can live an infective life cycle or a non-infective life cycle. And this is a great adaptation to have as an organism because you want to be able to use whatever resources are available to you at the time. So whatever opportunities it have, this fungus can carry out either life cycle and exploit nutrition from whatever option it has. And here's a closer look at that infective life cycle. So a spore will land on the insect's cuticle, also known as the exoskeleton. And spores are very good at adhering to insect cuticles because both the cuticle and the spore itself are going to have a hydrophobic texture. And because of that, they like to stick together. And once that spore has adhered to the cuticle, it will send out a germ tube, which is short for a germination tube. And this is basically the first strand of mycelium that's produced that allows it to be considered germinated. So it's starting to grow. And from there, it'll produce this structure called the aprosorium, which is this plug-like structure. And the aprosorium is very fascinating. It will begin excreting all these very specialized enzymes that allow it to penetrate that waxy, chitin-rich cuticle that the insect has. But it doesn't just use enzymes. It also uses turgor pressure. So it's like pumping fluids through this mycelial mass in order to physically push through the cuticle. And then once inside, it'll start producing more and more strands of mycelium, which will proliferate throughout the insect's hemocell, the body cavity. And this fungus knows to specifically target the non-vital organs first before moving on to the more important organs that will then kill the insect once they're damaged. And the reason for this is that, if the fungus were to kill the insect right away and then have continue growing through the rest of its body, it would then be competing with other pathogens and other bacteria and fungi that are now growing on this dead insect body. So very successful adaptation for this fungus to be able to target those non-vital organs first. And if you think about it, it also kind of allows for better dispersal, because if you're this little grub or whatever, and you're infected by this fungus, you're going to crawl some distance before you die, and before more spores are released. So this also makes sense how this fungus has proliferated throughout the world. So once the fungus has completely grown throughout the insect's cadaver, it will, throughout its body and then making it a cadaver, it kills it in the process. It will send out these spore-bearing structures back outside of the insect on the external to then disperse spores and continue the cycle. So then now we get more spores, and the spore could then continue the non-infective life cycle in the soil or find another insect to infect. And here's what that would end up looking like. So here is some examples of insects that have been killed by Metarhizium. And you'll notice that these insects are dead, by the way. They have this fuzzy green texture externally. And this is what those spores would look like to the naked eye. So there's no mushroom or fruiting body that is produced by this fungus. It's just spore-bearing structures with so these hairs that are producing spores, and that's what this would look like. So we get this fuzzy green on the outside of the insect. And Metarhizium, it's a genus of fungi. This is just one like group of species of fungi. And it's a very, very diverse genus. So it's known to infect many, many, many different kinds of insects. And some of them are known to be some types of Metarhizium, some species or subspecies of Metarhizium are known to infect very specific groups of insects, and we would call that maybe a specialist fungus, where it's specializing in infecting a certain group of organisms. Or they can be more generalist, where they're going to infect a wide range of insects. And something else to note, Metarhizium is not the only fungus that does this. This is just one genus of fungus. And there are many, many different kinds of entomopathogenic fungi that have evolved separately throughout many different fungal lineages. So I'm talking a lot about Metarhizium in this presentation, but just know there are many different kinds of entomopathogenic fungi that are prevalent everywhere. So entomopathogenic fungi are very diverse. And early on, Metarhizium was actually discovered pretty early, surprisingly. You might think, oh, this is like something we surely wouldn't have known about for a while, but no, actually, we've been using Metarhizium. We, as humankind, have been using Metarhizium for over 100 years now. So this guy, Eli Metchnikoff, he's known to have discovered a lot of early pathogens. That's kind of what he's known for. And he discovered Metarhizium in the 1870s. And he originally observed it growing on a wheat grain beetle, which is one of these pests here. And just within like a decade from then, he started using this fungus to manage as a biological control these wheat grain beetles throughout Ukraine. And they were pretty successful. It reported 80% mortality. So, and you can imagine the wheat grain beetle probably would have been much more significant of a pest then, with people probably storing grain in their homes more, or maybe not having a great facility that's airtight. So this would have been a very relevant application for this fungus to be used to help people, preserve their grain. So now, flash forward to recent decades, Metarhizium is used as a biological control for insects in many, many different aspects of agriculture. And currently, there are over 45 different products that use Metarhizium as its main active ingredient. So what that means is it's mainly Metarhizium spores, plus maybe a carrier agent, like a carrier powder or a carrier solution that is aiding in the application of the spores. But because the spore is the main infective unit that does the mode of infection, the spores are what's used in these biological controls. So what I mean by that is, if we were to propagate a whole bunch of Metarhizium mycelium, it wouldn't necessarily do anything. It's really the spore that performs this infective life cycle. So these products are mainly powdered spores or like a solution with spores. And they're usually used in most cases for managing plant crop pests. So therefore, these different products are usually either tilled into the soil to kind of boost the natural population of these fungi that would already be present in the soil, or they're applied, whether it's like dusting or spraying, onto the foliage of different plant crops. And Met 52 here is kind of the main popular product if you look on the market. And with this all in mind, with knowing that this fungus is already used throughout agriculture, kind of the main thing with integrated pest management when you're using any kind of biological control, but especially with these fungi, is that the efficiency of your application is always going to be dependent on what conditions the fungus favors, specifically for germination in this case, because with the spore germinating, that is what causes this infective life cycle, like I said. So whatever conditions the fungus favors for germination is going to be what we will want to induce to have it work most efficiently. So with that in mind, knowing that Metarhizium is already used for control of many different insects, you might be wondering, well, why don't we try using this for managing the mites? What would be stopping us from simply dumping all these spores in the hive and observing the mites die? Specifically, what conditions are different in the hive versus out in the soil where these fungi are naturally found? And the answer to that is temperature. So kind of what makes Varroa mites like kind of the bottleneck for this is that these fungi are not used to germinating in high heat environments like a beehive. So, we have this picture here from earlier with the bees regulating their cluster for the winter. And throughout that time, not just in the winter, throughout the whole time they're a functioning colony, they're always going to be regulating their brood nest to have this high heat environment between 32 and 36 degrees Celsius. So this is much higher than most soils. And therefore, while Metarhizium has been shown to have some effect on varroa mite populations, because it's not acclimated to this temperature, it's shown to not be nearly as effective as miticides that we would need it to be closer to in efficacy. So in 2021, researchers in the lab that I'm in here at WSU began working with Metarhizium to develop a thermally tolerant strain of this fungus to start using it as a biological control for varroa mites. So if you'd like to read the whole paper, this is the title. It's in Nature. But if you look up this title, you'll be able to read it. And I'm going to go over briefly some of the methods that they use. So first of all, there was kind of selection done just in the lab. And that starts with growing a spore. Here it's labeled as mitospore. That just means an asexually produced spore, just like the word conidia, but regardless, just a spore is either grown on agar that has hydrogen peroxide applied to it to give it oxidative stress, or it's grown on agar that has very, very low nutritional value, which would be giving the fungus nutritional stress. And the reason in either of these cases that we would want to apply stress to the fungus is that we're more likely to see some kind of genetic diversity appear, because we're basically inducing mutation by giving stress to these organisms and putting them in a stressful environment. And the reason we would want that is that because these fungi are asexually reproducing, they're not genetically recombining with each generation as much as a sexually reproducing fungus would. So because they're asexually reproducing, we can apply this stress and get more genetic diversity after. And hopefully with more genetic diversity, more likely to see a thermally tolerant strain appear. So they would apply the either stress method to the spore, continue growing that spore out on agar till it filled out and it had all this mycelium. And then they would collect spores from that, increase the incubation temperature, and then repeat the whole process. So after several generations, they were able to take this to the field, and essentially they continued growing these spores on agar and then inverted that agar plate on the top of a beehive here. So you can see this plate is upside down with spores facing down. And then they collected mites. Whoops. There we go. They collected mites from the bottom of the hive that had died and put them on agar. And you can see here, these dots growing on the agar are vegetative blooms of mycelium growing from these mites. So the fact that the fungus is growing out of these mites shows that it's very, very likely that that's what killed the mite, because the fungus would be present inside of it, and then putting it on agar like that would allow the fungus to then grow out. So seeing this fungal growth is an indicator that it most likely was killed by Metarhizium, unless like maybe some Metarhizium happened to be on it somehow and get on the plate. So they would collect spores from these plates that grew from the mites and then continue this process again. And they were pretty successful. So this is a good image to kind of show the success. I mentioned earlier that spores first produce a germination tube, and that's the first strand of mycelium. So that's what we're looking at here, is we're looking at these tic-tac shaped spores that some have germ tubes emerging from. So all of these spores were germinated at 35 degrees Celsius, which is on the higher end of brood nest temperature. And this top row of images are from spores from the Metarhizium cultures that were not given any selection. So these are spores that are acclimated to room temperature, versus on the bottom here, these are the spores they selected for that high heat environment. And you notice that the germination tube is much more prominent in all of these spores that came from that selected strain. So this is some very good visual evidence showing that these spores are adapted to this environment because they are able to send out this long, prominent germination tube. And these ones on the top, they're not as good. They're kind of crappy because they're not all producing germ tubes, or the ones that are there are not as long. So these fungi would not perform as well in a high heat environment when trying to infect an insect. So when I came to this lab and joined the work that was already going on this project, we were trying to grow the Metarhizium on brown rice and apply it into the field on live bee colonies. So we would do that by first growing the Metarhizium on a whole bunch of agar plates, like this one in this picture here. And then once it's fully grown and we see these green spores on the plate, we would go into our sterile hood space where we have this sterilized brown rice, and we would then inoculate that bag of brown rice with the fungus by just scooping out everything in that agar plate and dumping it in there and mixing it together and allowing those spores to disperse in the rice. And, of course, the reason we need to do this in a sterile environment is if we were to open that bag of rice just in a room, wherever, or even in the lab where we are in that picture, just not in that sterile space, we would be introducing all the different contaminants that are in the air to that rice, and it would inevitably rot or mold. Mold is a fungus that's in the air all over, and that's why things go moldy. So doing everything in a sterile space is necessary when you're working with fungi, because otherwise you're just going to grow the fungi that are in the air naturally. So this hood keeps everything sterile. And of course, we went through a lot of brown rice doing this, which means somebody's got to go to Costco and get our special fungicide-free organic rice for growing fungus. And once those bags of rice get fully green and dusty with spores and mycelium, and we would call that fully colonized with Metarhizium, it's been colonized with the fungus, we weigh out 400 grams of that brown rice that has the fungus on it and put it in a paper bag like this one here. And you'll see in this picture, we have a little spacer put on top of the hive that adds like an extra 2 inches over the top of the bag, just to give like a gap for that bag to sit in. And kind of the idea here is that the bees, they're hygienic. They don't like having debris in their hive. So they're naturally going to tear at this paper and tear it open and start interacting with the grains of rice inside that are all dusty with spores. And they're going to end up getting spores all over themselves and onto each other and cleaning each other. And the goal would be that the spores get dispersed throughout the whole colony onto all the worker bees and around the brood as well. So by placing this in there, we are relying on the bees to distribute it onto themselves. And here's what it will look like for us to put it in. We're basically just cutting a little hole in the bag to kind of initiate that that interaction for the bees to have. And then we would monitor the effect of this by using sticky cards that were placed in the entrance of the hive to sit on top of the bottom board to catch any mites that would fall. So this is just a paper card that we've hand coated in Vaseline and then put a screen over to prevent bees from getting stuck in there. And then we slide it through the entrance, and then we'll pull it out a few days later or a week later, however long we decide to do it in that experiment. And then we'll count the number of mites. So you can see here's a mite there. So oftentimes in research, you will take something to a certain level and try it, and it'll either work or it might not work. And if it doesn't work, the only logical thing you can really do is take it back a step and look at it a little closer in the lab, and try to get a better understanding before then going back out and taking it to the next level, to the field. So while we were seeing some results from the rice setup, we weren't seeing quite the effectiveness that we would like to see in order for this fungus to be equally as effective as a miticide that a commercial beekeeper might use. So we took a step back and did some of these in vitro assays in the lab, where we basically replicated brood conditions by setting up all these little centrifuge tubes and putting bee pupae inside of them before putting mites on the pupae that were either treated with Metarhizium or not. And the reason that we're putting pupae in the tubes and putting the mites on them is if we were to just collect a whole bunch of mites from a hive and treat them with Metarhizium or not, and then have them in like a dish, they would immediately die of starvation within like 24 hours. So in order to properly test this and know that our mites are dying from the fungus and not just from starving, we got to replicate the brood condition. So that's why we did this. And so to set this up, we had to, of course, collect all the mites and the pupae from brood frames. And this took a long time. We had to kind of get a whole bunch of people involved. And you'll see in the video here, we were able to use paint brushes to kind of twirl up the mites, kind of catch their little legs and drop them off in this jar. So if you ever need to collect Varroa mites by hand for any reason, using a camel hair paintbrush works very nicely. So once we had all the pupae set up in the tubes for the mites to live off of, and we collected all the mites and had them in our little jar, we treated half of the mites with a Metarhizium spore solution and the other half with a control solution. So the Metarhizium solution was just a sort of a tea, well, not really a tea because we weren't brewing it, but basically like a solution that we mixed a bunch of this very sporulated rice in and then filtered it out to get this solution that was full of spores. And the solution itself without the spores was water plus tween 80, which tween 80 is just an emulsifier. Basically it prevents the spores from clumping, and it's used in a lot of situations like this where you have like a powder that you want evenly dispersed through a solution, through a liquid. So you use tween 80. And then of course our control solution, we had to have that same ratio of water plus tween 80 because we want to make sure it's the fungus that would kill the mites in the treatment group, not just the tween 80, right? So here's what that looked like to make the spore solution. So I'm just shaking a bunch of these rice grains in the solution and then filtering it through some filter cloth. And you'll see what I get below is this really nice dark spory solution. And then my friend here is taking these mites and individually putting them on the filter paper, pouring the solution over them, and then scooping them back up with the paintbrush and putting them back in their tubes with the pupa. So half of these mites received that spore solution, and the other half were just given the control solution. And we monitored mortality every single day by looking at each tube and making sure the mites were still alive or seeing if they were dead. And any dead ones we removed and surface sterilized before putting on an agar plate to observe this fungal growth that could come from them and indicate that the fungus is what kills them. And you'll see down below here, some of them started growing fungus right away while others didn't. And that would maybe be indicative that those other ones were not killed by the fungus. Maybe they died of some other natural thing, or maybe they were part of the control group, so they were not given any fungus. But examining this fungal growth is what indicates that success. So especially this green growth in the middle. And we would usually see this around 5 to 10 days later. Oops. And our results from this, just looking at the mortality, was really, really significant on its own. So this red line here represents the mortality rate of the Metarhizium treated mites. And then this black line here represents the mortality of the mites treated with just the control solution. And you'll see that by day 5, we had over 50% higher mortality in the treated group. So that's pretty significant on its own. And it directly correlates with what we know about the life cycle of these fungi, because the fungus isn't going to kill the mite right away, right? It's going to take like 3 to 5 days or 3 to 7 days. So the fact that we see this striking difference around in this range of time shows that, yeah, we're pretty confident these mites were dying of Metarhizium regardless. So the question that is always on everyone's mind, understandably so, is how will this fungus affect the bees? And how do we know that, you know, if we're talking about applying these insect-killing spores to a beehive, what if it's just going to kill all of our bees that we want to keep alive? And that's an absolute valid question to have because we want to save our bees. We want to protect their health, protect their longevity, and we would not want to be developing some product that would potentially hurt them. And luckily, we know already that we're pretty confident, and we're going to continue to investigate to confirm that this fungus has very, very low effect on honeybees. So first of all, there are other labs who have done similar work with Metarhizium for Varroa mites, and there were some labs that directly tried to inoculate the spore solution into brood cells that had pupae in them, or brood cells that had larvae in them, and they also did the inoculation on adult bees as well. And basically, the Metarhizium was pretty much unable to infect adult bees. However, there was a slight mortality ratio, like under 10% mortality of larvae and pupae of honeybees. So there's kind of a few things to unpack here. So first of all, the reason that we're pretty confident that this fungus is not able to infect adult honeybees is two different reasons, and these are both traits that are very, very defining to Apis mellifera. First of all, that they're fuzzy, and second, that they're very hygienic. So when we see bees that are covered in fuzz, we know that they have this fuzz for the purpose of collecting pollen. And in this picture here, we see that all the pollen is being held away from the bee's body, away from its exoskeleton, because it's all trapped in this fuzz that the bee has. So it's likely that any spores that land on an adult bee that's fuzzy like it should be are maybe unable to germinate in a way where the mycelium is able to reach the cuticle and perform that, generate that aprosorium and do all that. So because the spores are probably being held away, away from the bee's exoskeleton, that germ tube's probably not long enough to reach. So that's one reason. But then also the fact that bees are very hygienic. They're constantly cleaning each other, cleaning themselves, and they have these different adaptations for, combing their hair and all that. Mites are not like that. So it's also very likely that any spores getting on these adult bees are getting cleaned off pretty quickly. And then the other thing to kind of unpack with the mortality rate that's very low with pupae and larvae is that if our mites are in our brood cells where these larvae and pupae are going to be, and that's the area that we specifically want to target, it might be acceptable that a low amount of our larvae die if that means that all the mites that are exposed at that time with open brood also die. So let's say you wipe out 99% of your mite population and 2% of your bee larva population, that could be acceptable in a strong bee colony. And another great thing about Metarhizium is that it poses very minimal health risks to people. And what I mean by that is that, it's not good to breathe spores or to breathe dust in general. So being around any type of fungi, you don't want to be breathing the spores. So we know that this fungus is not a threat to humans, and it's not pathogenic to humans. But we would want to keep in mind that we want it to be applied in a way where we're not going to be breathing in a bunch of spores, whether that's something to do with the application method or something to do with wearing some good PPE, something. So the next thing that we're going to be confirming is going to be the best modes of application with that in mind. So, we tried different things such as the rice application, and that would be an example of a very passive mode of application where we are relying on the bees to expend energy, tearing at that bag and moving everything around and cleaning everything out in order to get the treatment on them. So it's very passive. We are putting it in the hive and making the bees do the work. And that might be kind of comparable to, these are showing different oxalic acid application methods. The rice method might be kind of comparable to these shop towel methods that people do, where you're relying on the bees to do the work of interacting with the treatment. But there could also be some other options to explore that would be maybe more direct and require less work of the bees. So for example, with oxalic acid, we can vaporize a whole hive. Maybe we could do something with an aerosol sprayer and spray some spores throughout the hive, or maybe we make a spore solution that gets dribbled between the seams of the frames, something. So with all these different options to explore, there's we're still confirming which ones are the most effective and which ones are the best forms of application. But the goal is for this to be something that is available to help commercial beekeepers. So whatever application method ends up being confirmed as the best, it's got to be something that is very quick and easy to apply. So maybe that could be something that goes in the entrance of each hive for just 30 seconds or a minute, like how oxalic acid vaporizers work. And then a beekeeper could go around to each hive and do that. But, if it's going to be something that takes more than a couple minutes, a commercial beekeeper might not decide to go with that strategy. So it's got to be something that is quick and easy. And like I mentioned before, there are many, many, many different groups of fungi, different taxonomic groups, and different lineages of fungi that show this entomopathogenic capability, this ability to perform infective life cycles on insects and kill them in the process. So Metarhizium, we've talked a lot about today. It's just the tip of the iceberg. There are many, many different kinds of entomopathogenic fungi. So this is kind of the start to talking about my research that I'm doing, where I've opened it up to more than just Metarhizium, but also 2 other kinds of entomopathogenic fungi. And I believe that having more groups of these fungi to work with, more lineages to work with, we're basically opening up our pool wider to find some really virulent strain that's, virulent towards varroa mites, low effect on honeybees, and, has a high heat tolerance, etc. So by looking at more than just Metarhizium, I hope to find this solution. So here's the 3 fungi that I'm working with: Metarhizium brunium, Hersitella thompsoni, and Beauveria bassiana. And the Metarhizium and the Beauveria I purchased as cultures from this national culture collection that the USDA manages in Illinois. And then the Hersitella I received from, from the Extension Center over in Wenatchee, where they do a lot of work with orchard integrated pest management. And so they're working with these same fungi for the purpose of managing codling moth. So I was really excited to get this culture from them of Hersitella to start my work with Hersitella, because they specifically isolated this from a codling moth that they found in an orchard that was producing this fungus that had, so clearly it had been killed by Hersitella. So that's kind of just a cool fun fact of how that one was acquired. And here's kind of the process of what I'm doing with these fungi. So the first step with any type of selection of any type of organism is always going to be some initial screening. So in my case, when I'm screening for thermal tolerance, I will first grow all these fungi vegetatively at different temperatures, ranging between 27 degrees Celsius and 35 degrees Celsius. So, I have them all on trays in these different incubators. Not too exciting to look at, I know, but this initial screening kind of gives me like a baseline idea of how vigorous these fungi naturally will grow at these temperatures. And then hopefully later on, when I have these heat tolerant strains, I'll be able to compare this vegetative growth with how the things I end up growing at these high temperatures. So this not only gives an idea of baseline vigor at different temperatures, but also gives me a starting point to start selecting things. So after I do that initial screening, I start selecting single spores where I'm basically going in with a microscope and, I have this heavily diluted concentration of spores that I'm spreading on an agar plate. And then I continue incubating them at these various temperatures. And then I come back 16 to 20 hours later and look for these germ tubes. So, I've talked about the germ tubes a lot already, but it's a sign that it's germinated. And this gives me a way to measure the rate of germination. So I can basically say, 80% of these spores are germinated by counting how many have germ tubes and how many don't. And then from there, not only am I able to measure the germination rate using this method, but I will then take a sterilized needle and go in and just touch the end of one of those germ tubes and then touch it to another plate. And what that is doing is it's now inoculating the next plate with a little microscopic piece of mycelium from that single individual spore. So now I'm transferring a genetics from a spore that I know successfully germinated at a temperature to the next generation. So now I'm selecting those genetics that were able to germinate at that temperature. And so not only am I doing that, but I'm also, this gives me an advantage because if I'm selecting a strain, I would ideally want to have, at the end, when I have my strain, I ideally want to have some kind of genetic marker that would allow me to differentiate between my strain and any other isolate of this fungus. So by now continuing this selection method from a single individual spore, I know I'm working with one genome versus if I were to just continue transferring just a mass of spores, I know I'd have thousands of different genomes in there that I'm working with. So I can kind of observe the genotypic changes better by transferring a single spore, and then I'm also able to hopefully develop like a very defined strain. So those spores are transferred, and then they begin to grow out into mycelium, and this is what that mycelium looks like under the microscope. So we get individual strands growing together to create this sort of mat. And here's what that looks like up close. So hyphae is just an individual strand of mycelium. So mycelium is made up of hyphae. Don't need to think about that too much, but here's what those spores would then grow into, this web of these branching structures. And throughout this whole time, I am, continuing to select and incubate these generations at basically as high of a temperature that I'm seeing success at. So for some of my fungi, I started at 27 degrees Celsius, and now I'm seeing improved growth at 32 degrees Celsius, which is showing that there is a difference, but I want to continue doing more and more to try to get them to be thermally tolerant and able to germinate at 35 degrees Celsius so that they're truly not limited by the heat environment. And as I'm doing this whole process, every few generations, I have to make sure that I'm confirming that these fungi are still virulent, meaning that they still have the capability to infect and kill an arthropod or insect. So, when I am continually culturing these things over and over on agar generation after generation, there's a good chance that someone doing that could unintentionally select for a fungus that is now really wimpy and doesn't perform well outside of that environment where it's just in an agar plate. So to make sure I'm not selecting for a fungus that now no longer is able to infect insects or arthropods, I ensure virulence and confirm that it still has that capability by applying it to wax moth larvae in the lab. And the wax moth larvae, the reason that I go with those is they're very easy to rear in the lab. Sometimes beekeepers will, give me a little bit of crap for that. They'll be like, why are you rearing wax moth larvae? Isn't that a pest? And absolutely it is. But in the lab, away from comb and beekeeping equipment, it works very nicely for this purpose because they reproduce so rapidly. And also, they are often used for observing fungal pathogens because they melanize very quickly. So I'm able to start seeing these dark spots on their bodies as the infection spreads. And that kind of gives me a visual on how quickly they're being infected and if they are infective at all. So these are ones that would look like, that would already be completely dead and black, but you can imagine some of them might have just a couple spots or something like that when they're in the process of being infected. So here's an overall look at that process. I started with my initial screening, selected mycelium from that initial screening, and then continued incubating them at the different temperatures and selected individual spores that germinated successfully at the highest temperatures that I observed, and then confirming virulence throughout this whole process and repeating. Currently, I am in my third generation of selection, and I am seeing some difference, not enough to really have data to report, but I'm going to be continuing this selection throughout this fall and winter in order to gear up for a field trial next spring. spring 2026. So here's where I am in that timeline. A lot of this was because I'm in an entomology lab and I'm not in a lab that's doing work with fungi other than this project. I had to develop a lot of methods and comb the literature and figure out what different culturing techniques were already used for these fungi and how they had already been cultured for Varroa mites. So I started with developing methods, and then I acquired all my starting cultures from these different culture collections or from the Wenatchee extension, like I mentioned. And then I had that initial screening, and now I'm here in this strain selection period. So that's kind of where I am on the timeline. But then this spring, like I mentioned, spring 2026, I'll be doing a field trial and transferring these different fungi to the field trial. And in case it wasn't clear, all of these fungi, I'm growing them all separately on their own. So the idea is that over time, I can compare which of these 3 species of fungi are responding best to my application or to my selection methods, and which ones are ultimately going to be best for managing Varroa mites. So I'll be testing all 3 of the different species in the field trials, and then continue to confirm the best application methods as I do that. And then hopefully find a way to differentiate my strain from what I started with, whether that's a molecular marker or some other kind of observation with the genetic change. And then, of course, EPA approval has to come before this can be a product that is available on the market. But that's the ultimate goal, is for this to be a fungus that will become the main ingredient in a product that can be used on honeybees to kill Varroa mites, and can be used commercially and also used by hobbyists. And yeah, it would be the first biological control for Varroa mites because there currently is not one. So just to conclude a little bit here before I wrap it up, we know that honeybees are facing a lot of stress, and we've seen higher and higher rates of mortality. This is something everyone should be concerned about, and we need to find a solution urgently. And Metarhizium, we know it's already used as a biological control. It could be the first biological control for Varroa mites, but we need to first develop thermally tolerant strains. So by looking at more than just Metarhizium, I hope to find maybe multiple thermally tolerant fungi that I can create through these selection methods, and hopefully have some products pretty soon that will help beekeepers. So I hope that this was interesting, and I hope that all of you watching this have some new ideas of how fungi can be used to help our honeybees. So with that, I would like to acknowledge everyone in my lab who has helped with this project, and also thank WASBA for continuing to support and fund this research. I couldn't do it without the support. So, and then with that, I'm happy to take any questions or ideas that people have. And if you're, not wanting to share now, or if you think of one later, feel free to take my email and email me a question or idea later. And I also wanted to share these electron microscopy images that I imaged of these spores recently. So that's what you were seeing kind of interspersed throughout the presentation. But yeah, thank you. And I'm happy to take any questions.
Oh, thank you so much, Molly. That was really interesting. Are there any questions from the viewers? I don't see any right now. I was kind of wondering what you think the field trial will look like.
What I think it will look like?
Yeah.
Well, so currently we are still performing our field trials over in Moscow, Idaho, just because we require a special use permit in order to apply unapproved pesticides on honeybees in Washington state. So we'll be continuing to commute to Moscow, and I have not decided yet which application method I would like to test first. But I have a couple ideas in mind. And, at that point in time, I plan to have strains that are ready to go and that do show germination capabilities at 35 degrees Celsius or higher. So my goal would be to apply colonies with these fungi that I've selected strains of, and also apply some colonies with these same fungi, but from cultures that are from prior to my selection. And what my hypothesis is that the fungi I've worked with to select these thermally tolerant strains should have a much, much higher effect on mite mortality than the ones that have had no selection performed on them at all. So not only would this then be kind of testing in the field the efficacy of these fungi and how well they're able to kill mites, but also kind of demonstrating the effectiveness of my strain selection and show how having this thermal tolerant trait does lead to more success with managing mites. So that's what I have in mind for the field trial.
Okay. And so the Metarhizium, is that the fungus that Paul Stamets was seeing the bees use?
Maybe. So Paul Stamets, he did initially start our work here with Metarhizium. And initially our very first trials, like before I was involved with the lab, he was here and involved with that. But I think maybe what you're thinking of is he also separately did a lot of work with us with fungal extracts and seeing if they would maybe prime bee immune systems when we were feeding the bees the extracts in sugar syrup. So kind of different. The fungus isn't killing anything, but hopefully priming their immune system. And that was inspired because he at one point observed bees feeding on wine cap mushroom mycelium in his garden. And so kind of the thought with that was like, potentially they could be, you know, extracting some kind of like moisture from the mycelium that maybe is giving them some kind of like nutrient value that then we could replicate by doing the extract thing. So I think that might be what you're thinking of, where he observed them interacting with that mycelium.
Probably. There is a question. Is there a way to maintain purity of the strains you select?
Is there a way to maintain purity? I think there's a couple different ways I could interpret that question, but I think what this person means is probably like, well, so something to consider is that fungi change very, very rapidly. And like, while we're used to looking at mammals where they will, you know, mate and reproduce into offspring, and then that's the next generation. Fungi can technically be kind of producing many different generations on their own in a single plate. So there could always be potential that a thermally tolerant strain could be selected for and then potentially unselected for if then that strain wasn't maintained in a high heat environment. So there's a lot more to learn about this, but so far from what I understand, we would need to maintain purity of this strain by continually propagating these spores in that high heat environment and then using those spores to then go into a product. So, you know, if someone, for example, were to try to grow those spores and then they grew them at room temperature, it could then be reacclimated to room temperature. So when this does become a product that people are using, there's going to have to be some kind of facility in place where people are, you know, whether that's me or somebody else, is going to be continually growing this fungus in a high heat environment and then only using spores from those cultures then in the product.
Okay, we have another question. Under very good conditions and a lot of luck, how many years do you think it would be for a product to be actually available through the EPA?
That's a good question, and I get that question quite a bit. And I almost don't even want to like throw out an answer just because I'm just so unsure of how long it could possibly take. But really, kind of the main time-consuming thing with that is just getting enough evidence and backup data in order to get that EPA approval. So we could find a thermally tolerant strain that's very effective and we find a way to apply it that's really well. But that EPA approval is really kind of the secret sauce that we need in order to get this as a product on the market. So that can take years and years, or it can take, you know, only a couple years. It really depends. And, you know, a lot of the stuff with like doing the paperwork side of things, it just kind of takes a long time. So like, for example, I mentioned how we need a special use permit in order to test these things in the state of Washington because it's an unapproved pesticide. And just getting that special use permit, you know, has taken so long and it's still pending at this time. So based on that, I don't have a very great judgment of how long it could be, but it potentially could be several years before this is a product, but hopefully sooner than that.
Hopefully. Do you think that this would have any application against other pests besides Varroa? At the WASPA conference, we saw a very kind of scary presentation about Tropolaelis. I can't say that mite, but the mite that is a concern.
Yeah.
And
Yeah. So with the idea of like, creating this fungus that is thermally tolerant and performs well in at beehive temperature, there's no reason that this fungus would not also be great at controlling or managing Tropolaelaps mite populations. Um, the only, in fact, I think that developing this product or this treatment strategy now, it could potentially be a really, really positive thing for everyone with preparing for Tropolaelaps if it does start showing up here in the US more. So, if we have this product going and it gets approved and it's effective against Varroa mites and people are using it, that might be a really, really good strategy to have in our tool belt if this Tropolaelaps occurs. So, yeah, I think that this very much will be relevant for that issue. And other than that, the only reason that maybe it could maybe not be effective towards Tropolaelaps is if somehow it develops like a specialist sort of virulence towards Varroa mites. But in that case, we could then try culturing on Tropolaelaps, and there's no reason we couldn't develop a strain that is specialist towards Tropolaelaps instead. So I think this is going to be a great strategy for if we do have to deal with Tropolaelaps over here.
terrific. And obviously it works on wax moth larvae. So
Yeah.
that's another, I guess hive beetle might be another one that we would want to see fungus work against.
Yeah, that would be
So,
interesting.
yeah. Are there any other questions from the audience? Well, it doesn't look like it. So, Molly, thank you so much for sharing your research with us. We're really excited that this might provide some relief for beekeepers in regard to the Varroa mite. And maybe next year we'll have you back to see how your research has progressed through the field trials.
Yeah, that would be excellent. I would love to give an update.
Okay, well, thank you very much. For those of you who are online, our next Ask a Washington Beekeeper will be the third Thursday of November, which I believe is the 20th of November at 6:30, and we'll have someone speaking about the use of pesticides and its impact on bees. So I hope that you will join us then. Thanks again, Molly.
Thank you.
And we will talk to you next year, probably. Thanks again.
Awesome.
Bye-bye.